Thank you very much, Dr. Hurd. It's really a great pleasure to be starting this afternoon session. The idea here is to really set the stage and show examples on how basic and translational research are feeding preclinical and clinical research. And rather to be trying to be comprehensive, I would like to show you examples on that, how this has been done, and also to pinpoint on some key aspects that are very early in the molecular design that could lead to inequalities. I'll start here with the general biology of cancer and recall how complex the cancer biology is. Here we focused on this review on just one cell type, macrophages, that are very numerous in some cancer types, and to try to understand what are the interactions of macrophages with different cell types, what are the states of macrophages, and how can we use that therapeutically. And you can see that it's a bit small to read, but we can deplete macrophages, prevent them entering the tumor. We can reprogram them once they are in the tumor, or try to enable their tumor killing capacity. So clearly just one cell, and we already see the complexity of this ecosystem. And now we have to factor in the number of cells, the different state of all these cells and all the interaction. So clearly a formidable challenge to understand all this. So now omics techniques and also the capacity to bring this omics into the spatial component, the so-called spatial omics, are really bringing a new dimension to this. And I would like to illustrate some of this in the slides to come. To understand this, it's very nice to have also a framework. And Doug Hanahan has provided a very interesting way to look at this as hallmarks of cancer. What are the key features that a cancer cell or a cancer tissue needs to be able to achieve so as to become malignant? And you can see some example here, being able to sustain blood vessels, to sustain proliferation, and so on and so forth. And you can see that for each of these hallmarks, there is a therapeutical intervention. So this is a very nice way to go forward in trying to combine hallmark-specific intervention or to combine several hallmarks in order to do combination therapies. And what is very important here is the new hallmarks that have been identified, and one of them is avoiding immune destruction. So it appears now that being able to go below the vision of the immune system is an essential feature of tumor cells. They need to be able to achieve this in order to grow. And that's changing the way we look at immunotherapy. Immunotherapy is not necessarily boosting the immune system, but it's actually restoring the capacity of the immune system to be able to eradicate tumor. And that's exactly the strategy that was followed by the two Nobel Prize winners, Jim Allison and Tasuku Honjo, who tried to really address this question on immune escape and how can we restore the full function of the immune system. Now we have very clear idea of what is being recognized on the tumor cell. You can see here the small green dots that are actually mutations in some small peptides that are presented to the effector cell, the CD8 T cells. These are the lymphocytes that kill tumor cells directly through engagement of the T cell receptor, which is this TCR shown in yellow. And clearly, there are several types of antigens that are being recognized on tumor cells, but the most important one probably are neoantigens. Those are mutations that are characteristic for the tumor and are read by the immune system for destruction. And what I illustrate here is a very famous protein called P53. And you can see that the mutation is shown here in green. And now you see the very same part of the protein being presented by this small presenting molecule called MHC, HLA-A2 in this case, to the recognition by the T-cell receptor. So we can really see how cancer needs those mutations to have this aggressive phenotype. And at the same time, this is what is being recognized by the immune system and is a way to get rid of the cancer cells. So this has been some of the prior work to the work of Tasuku Honjo, trying to now understand what happens when those T cells that are armed and enter the tumor. And what we know is that the tumor can actually protect themselves by expressing so-called PD-L1, and this small molecule actually blocks the action of the lymphocytes. And so it's a natural way that the tissue have to prevent the action of the immune system. And since the tumor cells are actually healthy cells that have evolved the wrong way, they still have this capacity to block the immune system through this PD-L1 expression and then triggering PD-1. So we can actually block this with monoclonal antibodies and actually restore this immune system. There is another case which is quite interesting, is when the cancer cell can directly, through oncogenic rearrangement, express PD-L1 at a high level, and that's also a way to directly escape the immune system. And I will show you an example where we have used this therapeutically in one patient in a few moments. These have been a fantastic success in oncology. You can see here the survival curves of melanoma patients. These are metastatic patients. Those patients will not be alive at 5 years. Probably none of them would be. And now we are reaching above 50%. So it really shows that this has completely changed the landscape. And we are also starting to see a cure. Some patients probably will never relapse from their melanoma because some of these are really not treated anymore. So can we go further? Now we have fantastic technology. One of them is really immune engineering. And that's an example here with a drug that just made it to approval, Tebentafusp. This drug is recognizing, again, an antigen on this small presenting molecule, HLA-A2. But at the same time, on the other side of the molecule, it's triggering activation of any lymphocyte through the CD3 protein, which is ubiquitous in the lymphocytes. And that's a way to basically activate any lymphocyte that is in the neighborhood of the tumor. And that's a very elegant way to trigger local immune activation and tumor cell destruction. And you can see on the lower graph that patients treated with this drug now have a much better survival. That's the orange curve compared to the blue curve. So we are reducing by half the risk of death into these patients thanks to this type of action. Now, if we move forward, we can also do engineering of cells directly. And that's an example here that I wanted to show because you can see the date of the paper. It's 2002. And the time it took, so we are now reaching the clinic with this model. The time it takes to bring this to maturity, we used computer simulation to optimize the sequence of the receptor in order to bind better to the tumor cell and to be more active in killing it. And so this in silico, this bioinformatics calculation took place 20 years ago, and now we are entering into a clinical phase. And so that's also something we have to work on to try to shorten these pathways so that we can get to patient much quicker. We have now mouse models that can show that the mice that have these engineered T cells have a much better survival. This is the orange curve that you see at the bottom compared to those that have the natural T cell receptor. So clearly here, T cell engineering has allowed us to improve survival into mouse models, and we are now moving into a phase 1 clinical trial in the few months to come, hopefully. Now I'd like to just jump into a very new and exciting venue, which is how to use these large-scale analytics to drive a patient assignment into treatments. And now we have a wealth of information due to real-world data. We can use clinical data, patient-reported outcomes, genomics, and also images that are playing more and more a crucial role. We have to integrate these data into a coherent system and then be able to bring them into a tumor board where we can make decisions on how to treat the patient. I think we are working very hard in Switzerland to be able to bring the information of that very same patient that we treated through the tumor board into the data set and have a system that can learn as we go with the new cases. We are now moving into a prospective phase with multi-omics. You can see the two lower panel where we do fresh tumor biopsies, and there we do omics, either targeted or exploratory, bring this into the molecular tumor board for educated decision on how to treat the next patient. And you can see here that there is a lot of spatial omics, imaging, mass cytometry, digital pathology, spatial transcriptomics. All of these will be included into our decision making. We have a tumor board which is running and using this type of information, and you can see the pathway. We use the material on the left, and then we have a lot of different specialties thinking about the biology of that tumor, using a lot of AI and data science to bring this to a conclusion and what would be the best treatment for that patient. We started this program in Switzerland, and that's an example here of our cluster between Lausanne and Geneva, where we have more than 3,000 patients that have been treated. And we see an interesting response rate between 20 and 30%. We treated all comers, and we have now a very good data set that we can learn from, because all these patients have received this deep analytic and a treatment. And that's just an example of a very young patient, 30 years old at the time he came to us. He was progressing fast on chemotherapy. It's a sarcoma that is known to be resistant to immunotherapy. But on the analytics, we spotted an amplification of PD-L1. It's exactly the case that I mentioned at the beginning in the lower panel. So we can see that this amplification spotted the idea that perhaps this tumor, despite being resistant to immunotherapy, that specific tumor needed an active way to escape the immune system. Hence the idea to give to this patient PD-1 or PD-L1 treatment, which we did. And the patient who was rapidly progressing on chemotherapy became a full responder on immunotherapy. So this is an example where these deep analytics really opened a new possibility of treatment for the patient, and which really did change the trajectory in terms of its oncology history. So based on what I've just mentioned, can we spot some early inequalities in these programs? I think for checkpoints, it's fair to say that we have now very good evidence that most ethnicities do respond fairly well to these treatments. There might be some subtle differences. We don't have high-powered studies to really conclude there. And I think the important question is really to make sure that the clinical trials that we're designing encompass these differences so that we have information as soon as possible on these aspects. Now, for the other two program that I mentioned with immune engineering, I think we have to be here extremely clear with the fact that we are using targets that are associated with this HLA-A2, this MHC molecule, that are really specific of the patient and its ethnicity. You can see that HLA-A2, which is really mandatory for these drugs to work, is only 40% of the US citizens, but 25% of the Romanian, and only 2% in Thailand. So we can see that here, directly at the molecular design level, we are creating a very important imbalance in the patients that will be amenable to this treatment, yes or no. So I think these really are important aspects to take into consideration. And that's, of course, also the case for the T-cell, the engineered T-cell program that I mentioned. Finally, for the program on precision oncology, I mentioned that there is a lot of data science and AI, and there are clear biases in case your training set doesn't encompass all the ethnic diversity, with the risk of your AI system not being reliable on patients that are not captured by your training set well enough. I think this will be discussed further in the session, but it's also something to point out as a limitation and something to look after carefully when we are building the training set. So finally, to conclude, I think we are now witnessing a very interesting period in oncology, where we have fantastic tools to go faster from the biology to treatment. These programs have the risk of introducing early biases into what patient at the end of the day will be candidate for treatment and could benefit from it, yes or no. And I think echoing what has been said in the session this morning, I think we really need to be able to factor that very early in the molecular design of our therapies, so that we can encompass as broad as possible the number and the ethnicity of the patient that we can consider. So with this, I'll stop here, and thank you for your attention.